Hey everybody, for this week's lab we're going to be measuring respiration rates in aquatic and terrestrial ectotherms. So ectotherms are animals that have a body temperature that corresponds with the temperature of their environment. So in other words, we're going to be measuring respiration rates in fish and in reptiles. And for this week's lab, there are a couple of pieces of instrumentation that I want you to be a little familiar with before we dive in and get started for lab. So let's get started on first talking about respiration rates in fish. So for studying respiration in fish, we're going to be working with two different types of aquarium water. Regular aquarium water that's had air bubbled through it, so it is normoxic, so it reflects water that should be at equilibrium with the air in terms of its oxygen content. And then I'm also going to prepare a tank of water that is hypoxic. I'm going to do that by bubbling some nitrogen gas through the water for long enough to displace the oxygen that is in that water. So one thing that you're going to wind up needing to do is you're going to wind up needing to measure the oxygen content or oxygen concentration that is in each of these solutions to examine fish responses to them. I'll also note with the hypoxia, I'm not perfect at getting all of the oxygen out, so that's part of why we need to quantify how much oxygen is left. So for doing that, we're going to use a pretty simple tool here. This is a dissolved oxygen meter. And I've gone ahead and I've calibrated these units already for you, so you don't need to do any of that. But what I do recommend is these usually work better if they've had a little bit of time to warm up and equilibrate. So when you come in, just make sure that you turn it on. And then while you get it ready, just leave it hanging over the edge of the lab table. That's really the ideal position for it to equilibrate. Okay? And then when you are ready to use it to measure the oxygen concentration in your water, first thing you need to do is there's a cap here over the top of an electrode. And just for your reference, inside of here is a little chamber that's filled with salt water, and there's a little mesh membrane here over that chamber, and that mesh membrane is going to keep the salt inside. And that's what's actually going to be measuring your oxygen concentration. So try to avoid coming into contact with or damaging that little membrane. So when you're ready to measure the oxygen content of your solution, all you need to do is put that into your, this is funny looking at, put that into your solution, and you'll notice here that's starting to tick down. And so it may take a minute or two to reach a point where it will settle. Looks like we're getting close. So just be patient with it. Wait until it settles. And it actually uses oxygen as part of its sensing mechanism, so it may not completely settle. So you just want to wait until it reaches a relatively stable reading. So you're going to take that reading, and that's going to be in milligrams per liter. Okay, so that's my normoxic solution here. Okay, so let's just compare what that looks like relative to my hypoxic solution. So here's the hypoxic solution. And I'd note, so this is giving us a concentration of oxygen, right, in milligrams per liter. So one of the things you're going to wind up needing to do when you report your data is you're going to need to convert this to the water oxygen partial pressure. And we'll walk through some of those calculations a little later on. But as you can see, so from bubbling that nitrogen gas through, my dissolved oxygen concentration in this hypoxic water is a lot lower than it was in the normoxic water. So we're going to be able to directly test something I told you about, which is how fish respond to changes in environment oxygen availability. I forgot one other thing I wanted to add is we're going to work with two different fish species, both common fish species. So we're going to work with some betta They're a little bit small, so it may take a little bit of time to figure out how to quantify respiration rates. We'll just be measuring on opercular openings, so openings of the gill covers. And then for our other fish, we're going to work with some goldfish. The goldfish are a little bit easier to see, so it may be easiest to start out working with the goldfish before switching to the beta. Along with that, we're also going to characterize thermal responses in the fish and in reptiles. And so for that, with the fish, I've got a set of aquarium heaters that are on each table. And the main thing you need to be aware of with the aquarium heater is don't plug it in until you put it in water, and don't leave it running out of water. It's not going to cause horrible problems, but last semester we did have a group that accidentally left it running, and some plastic melted onto it and it started making some smoke, and it was a little exciting. So just general rule of thumb, put one person in charge of the aquarium heater, put it into the water, then plug it in and turn it on, and unplug it before you remove it again from the water. Okay, another thing with measuring temperature, so we're going to put the fish into these beakers, so there's a nice volume of water to work with. You'll get at the appropriate time, you'll get your aquarium heater going. And one challenge is that just doing this, it's very easy to wind up with a thermal gradient in your beaker. So the other thing that you'll have on your table is a set of stir magnets. So put one of those stir magnets in the bottom, and I've got a little stir plate here as well. So you don't want to send your fish into a whirlpool with a stir plate. So just get it set up and start at a very low stir rate, and it generally should be able to keep a pretty low stir rate. You want to keep that constant across your different treatments. Okay, so those are our fun projects with fish. As I was mentioning, the other thing we'll do is we're going to measure respiration in our reptiles. And for doing that, we're going to go back to using our Power Lab units again. And this time, instead of using the finger pulse transducer, we're going to use a different type of sensor transducer, which is one of these force transducers. So the way these work is, obviously I've got the business end here to plug into the Power Lab unit. So it's got this set of blades, and you'll notice there's the top portion of the transducer is the part that has the label on it. So it kind of has this angle to it, and then the other side is going to be the underside of the transducer. And these blades here, there's a set of holes at the end. And depending on the magnitude of the forces you're measuring, that's going to determine how many of these blades you use in kind of detecting the signal. And the principle here is pretty simple also. Basically, changes in the bend of these blades is going to translate into a change in voltage that's going to be detected by the Power Lab unit, and it's going to give you a measure of force. For measuring reptile breathing, the reptiles, that breathing rates are a comparatively small force. So we're actually going to move most of the blades. They'll just rotate out of position to the side, and we're just going to work with the thinnest blade here. And you can even see that that will start to bend from my finger weight. And one other quirk with these is these are not OEM instruments to work with the Power Lab, so our startup sequence is actually going to be a little bit different with these. So if I go over to my Power Lab unit, in this case, what I'm going to do is I'm going to turn on the Power Lab. I'm going to wait for the green light. Then you have to look at the tip of the plug to get this oriented in the correct direction. But once the power is on, the other thing you can look for is there's a little channel in the top of the plug. So you can see that there. That little channel tells you that's the top. So you can then, once Power Lab's on, plug in the force transducer. And then once that's plugged in, then you're ready to fire up chart. Okay, so as I'm getting that all fired up, then the next thing I'm going to do is I'm going to put this transducer into this ring stand, right, making sure the top part is up. And for right now, I'm just going to clamp that into place. Okay, so I'll stay in place right there. Okay, so then I'm going to fire up chart. I'm going to dismiss terrible updates. Okay, and then within chart, there's a couple of settings that I want you to make note of that are going to be different. So we're just going to start a new file. All right, so here I am in Power Lab in channel 1. And the first thing I'm going to do is I'm actually going to change the input voltage range. So that's over here on the right. Right now it's set to be 10 volts. And if I click that, there's a drop-down menu. And what I want to set that to at this point is the 50 millivolt range instead. So it's going to be detecting across a much smaller voltage range than we used for the pulse transducers. And then along with that, we're also going to adjust our sample rate. So this is the input voltage. Right above that is our sample rate. And we're going to change our sample rate to 200 samples per second, right? The default is 1,000 samples per second, so we're just going to go up to 200 samples per second. Okay, so now I've got everything set up, and I've just repositioned the transducer so you can see it when I press record. So I'm going to push record. Okay, so it's a slower sample rate. And it's actually automatically rescaled my axes. And now let's just watch as I tap the tip of the transducer. All right, so it's detecting changes in force as I tap the tip of the transducer. And I'm just doing that as a way to test the transducer, make sure that it's working as I expect it to. Okay, so now the next thing we need to talk about is actually getting your reptile hooked up to the transducer. And we're also going to be looking at temperature responses in the reptiles. And so to do that, we're also going to work with a set of probes. Okay, so for measuring reptile body temperature, what we're going to do is I'm going to take a set of these thermocouple probes. I have a set that I have specifically set aside for this purpose. So I'll be helping you with this setup with your reptile. And basically what I'm going to do is I'm going to take a little piece of, this is just dental wax, and I'm going to use it to kind of make a little tip on the end of this probe. And then the best way to measure a reptile's body temperature is in that reptile's cloaca. So the cloaca is an excretory organ. Unlike human excretion, the rectal muscles are not as constrictive around the cloaca, so it is not as uncomfortable as it might seem to us. But on the other hand, nobody really likes having something put in their cloaca. So what I'm going to do is I will insert this into the reptile's cloaca for you, and then I've got some vet wrap to wrap the reptile's tail to hold the temperature probe in place. And depending on the reptile, sometimes this can get to be a little bit squirrely, so we're just going to have to be patient with that part of our setup. Okay, so I'm not going to go through a full demonstration of this, but here is Gary, our awesome Garrosaurus major. He's doing a little bit of shedding, and he's a little annoyed at me for waking him up from a nap. But just to kind of give you a sense of what this is going to look like, I'm going to turn him over, and then right here is Gary's vent, and so I'll be putting the thermocouple probe in right there. And after I do that, for measuring breathing rates, what we're going to do is we're going to want to secure our reptile in place. And Gary's really great about this. Usually he's pretty happy to just chill. So we're just going to make a little harness out of some gauze and some tape to hold him in place. And then the next thing that you're going to want to do is you have a toolkit on your table like this. And in this toolkit, there should be a spool of thread. And you're going to take a length of thread. And what you want to do is get, kind of a nice big slip knot in your thread. So make sure someone in your team knows how to tie a slip knot. You might want to Google that in advance. Right? Okay, so you're going to tie a slip knot in your thread. And then you want to keep your loop of thread pretty big so you can fit that over the reptile's head and around its rib cage area. Okay? Then once you're around the rib cage, you can gently snug the slip knot. Let's see if I can show you down here. Okay? So you can gently snug this slip knot. Okay? And then once you have that together, then you're going to take your reptile and your thread. Okay? So you're going to take your thread and you want to thread the thread through the hole on the force transducer. And it doesn't have to be super tight at this stage. You're just going to want to secure it to the force transducer. Okay? And then the trick for measuring your reptile's breathing rate is what you want to do is get enough tension on the thread that you can measure changes in force production, but you don't want so much tension you're pulling on your lizard. So what we found the easiest way to do this is you can kind of do two things. You can adjust the height of the force transducer, or you can kind of move your tray closer or further from the transducer. So like right about there is probably pretty good height. And so now I am ready to start recording Gary's breathing. Let's see if I'm getting a detectable signal yet. I am. It's very tiny at this stage. But what you see there is Gary breathing. So let's try an auto scale. Okay, hopefully you can see that. Maybe I'll move you a little closer. Let's take a look. All right. Okay, so that's the basics for setup for our lab this week. With the reptiles, we'll be doing kind of some similar things, measuring temperature responses. So I have a set of heating pads. The heating pads can go underneath the tray, and you can also drape one across your lizard to warm your lizard up. And then I also have a set of Ziploc bags and some ice so you can cool your lizard back down as well. Along with that, I'm hoping it's always a little tricky to get to this just because of the setup for this lab. But I'll also have some bags that are filled with carbon dioxide gas, and we're going to use a little pump to blow gas very gently across reptile faces so we can also quantify responses to increased carbon dioxide levels. This is kind of fun. You can see Gary continuing to breathe here. Thanks, Gary. You're the best. All right. I hope you're looking forward to lab.